Hi, everybody. Hi. That was actually kind of easy. Okay, cool. Hi, everybody. This is an incredible turnout. I'll be honest, I wasn't expecting this many people to come. This is amazing. Perhaps if it, be a little bit squishy today, so maybe we'll have to look into, expanding the room. So stay tuned just in case if there's some room changes. But at this point, we'll be in this room. I'm so glad so many people have found it. Before we really jump into anything, I just wanted to again remind, you probably heard me say this 500 times now, and those my colleague Alexander, who's just over there, to please complete the attendance form. This is just so we can track who's here, so that we can issue you certificates at the end of the program, assuming you attend enough of the sessions. So again, you can scan the QR code here. There's also some paper forms just at the front. You don't have to complete both of them. There's a chair kind of in the back over there. And on the other side too, on my right hand side, the left side of the room. If you also haven't received a lanyard, some of you have like the ORT red and blue lanyards around your neck. We have a ton at the front, so feel free to come grab some at the end. We have lots, so there'll be some there. Maybe we will need to look for another room next time. So again, stay tuned. So before we begin, I think it's really important for us to start with the land acknowledgement. So for thousands of years, this has been the traditional territory of the Huron-Wendat, the Haudenosaunee, and most recently the Mississaugas of the Credit River. This territory was the subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Haudenosaunee Confederacy and the Confederacy of the Ojibwe and Allied Nations to peaceably share and care for the resources around the Great Lakes. Today, the meeting place of Toronto is still home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work and learn on this territory. I'd like to encourage everyone here as well just to reflect on the land which you all individually occupy. Come on in, you may have to squish and maybe set up some stairs, but we will figure this out soon. I was fortunate enough to grow up and do all my research training and education in Manitoba, so that Manitoba land has had a significant impact on my own journey. So again, I encourage you all to do the same as well. But as you embark into your research here at UHN, I just want to encourage everyone here to review Canada's Truth and Reconciliation calls to action. These calls to action were presented in 2015 by the Truth and Reconciliation Commission after 6 years of research and community engagement on the terrible impacts of the residential school system in Canada. These calls provide guidance on how each and every one of us can work towards reconciliation in Canada with Canada's Indigenous people, including recommendations for changes in the healthcare system. So us as researchers, it's really important to us to recognize the harmful ways that Indigenous people have been exploited in research and learn the best practices for including Indigenous people and Indigenous knowledge into our research wherever possible. Now, so welcome everybody to session 1 of the STAR program. As I've said already, it's fantastic to see so many people in this space. I do see a few empty seats kind of popped through should you want a chair. Again, like I've said already, we'll see if we can find another space that will accommodate everybody, because again, this is probably some of the best attended summer program sessions we've had so far. So it's wonderful to see so many people here. So I guess before I just jump into some of this, today we'll really be talking about, so the first half of the session you'll be hearing from me talking, so about 30 minutes, and then we'll passing it over to some of our great graduate students and postdocs. We'll give you an overview of what an idea or a sampling of what research looks like here at UHN. UHN is such a huge place and such a diverse place. So in research, every little bit of research is different. So we wanted to highlight all that for you today. So as you may have noticed, this presentation is being recorded. So if you're not able to attend in person, that's okay. We have the recording that'll be made available to you in an email after the session, and also the slides when that's relevant. Please feel free to ask questions at any point. Raise your hand. We'll also, Alessandra and I will stick around after the session. So if you have any questions about anything, please feel free to let us know. And as well, this is a safe space, so we encourage that everyone follows the ORT's Code of Conduct and Inclusivity Commitment. We have a QR code to that just on the screen, should you like to review that. Okay, so as you already know, you are here for the STAR program. So this is as you all probably already know, it's a six session program where you'll learn all kinds of different information on research skills and professional development. It'll culminate with a research day. We'll share all of that information about research day in June, as well as how to register for it, all the guidelines for participating. As well, by the end of the session, if you attend and participate in 4 out of the 6 learning sessions and participate in research day, you'll receive a certificate of program completion. Come on in. I know there's like no chairs, but you can come closer too if you want. I just want everyone to see. So again, there are 6 learning sessions part of the program. This is just a quick overview of the different schedules. So you'll be receiving emails from me before and after every session, including links to the recordings and information about where the session will be. Typically it's in here, but again, there's so many people here. So again, we'll all maybe look for a different space. But these are the dates here. It's located on our program website. And again, they'll be shared with you by email. At the end of the program on August 13th, there is a research day. Just to reiterate again, all that information will be shared to you throughout the summer. In June, how to register, and then we'll have a learning session on July the 10th, where we'll be able to go through tips and tricks on creating a poster as you prepare for research day. If you've missed a session, that's totally okay. Every session is recorded, and that'll be shared with you after. Part of the certificate requirement is that you attend or watch 4 out of the 6 learning sessions. So you can watch the recording and then complete what we call the missed sessions form. Just a Google format, you can indicate and note 2 things that you've learned about the session, and then that'll count as if you were sitting here in person. So no worries if you have to miss some learning sessions. There's no minimum number or maximum number that you can watch. If you happen to watch the next 5 out of the 6, that's okay. All right, so just to giving you a little bit of an overview of research at UHN. So research at UHN is conducted within the research institutes, and it spans a full spectrum of different disease areas and disciplines. So this includes things like cancer, cardiovascular sciences, transplantation, neural and sensory networks, musculoskeletal health, rehabilitation sciences, and as well as community and population health. So then this picture here is just depicting a number of the different research areas. And the research is organized across 2 research institutes. There's the Princess Margaret Cancer Center, and then there's also the UHN Research Institutes. Just to kind of put some clarification behind this, UHN is also in the process of restructuring. So the 2 research institutes, it's its new kind of organization of research institutes. Before, it was organized into 6 research institutes, so the Princess Margaret Cancer Center, as well as the 5 below, which now make up the UHNRI or the UHN Research Institute. So you may hear some of this language, either both of these different languages. So just to avoid some of the confusion, it's an evolving time for the research organization. But regardless, you're part of UHN, you're part of UHN Research, and it's an exciting place to be. With that, I'd love to kind of gauge the room a little bit to see where people are from, whether you're in one of the 2 research institutes, maybe part of an affiliated department, an administrative department or a clinical department, or maybe you just don't know, and that's totally okay. So I'll give like 2 minutes to do that so that we can keep on time. There's also a couple chairs in the back on this side. Okay, it's amazing. A lot of people from UHN Research Institutes, which what I expected, but people from Princess Margaret and from other departments. It's nice to see it's nice to see maybe nobody, and I'm not sure, which is cool. All right, so we'll just keep going. I just want to give you again some tips and tricks on just some resources and getting started at UHN, advice on maybe some of your the mentor-mentee relationship. And then again, we'll pass it over to our grad students and postdocs. So To help you get started at UHN, the ORT, or my office, the Office of Research Trainees, has a student and postdoc handbook, and this has tons of information about getting started at UHN. Well, within this document, there probably is a lot of information geared towards graduate students and postdocs, which might not be relevant for you. There still is a ton of information that will be helpful about getting started at UHN. So there's information like contact information. So if there's any questions about, payroll or research services, supports, departments, anything like that, this is a place where you can find all that information in one spot. There is information about getting connected at UHN. So find things about like getting network access to the staff shuttle. Some of you may have taken that if you're coming over from the Toronto Western site. Research directories, access to the library, events calendars, and a whole bunch more. There's also information about different research facilities and services, whether or not your research will involve outsourcing some of those departments, policies. Those are great things to have kind of access to should you need. Different groups at UHN like research integrity, commercialization, the IDEA or the Inclusivity, Diversity, Equity, and Accessibility Department, as well as we have links to different career and professional development resources. So a huge document that's great for you to review if you have some time. You'll be able to find this document as well on our website. There's also a QR code at the bottom. Again, I should just mention any QR code or any link that we share, I'll include that in an email after, so no need to capture those links or take a picture really quickly. But again, we also recognize that onboarding and getting started at UHN is overwhelming and there's a lot of things to do. So if there's any questions, Alessandra and I will be here after the workshop and either direct you to the right appropriate people or help troubleshoot some of those problems. Or if you have any questions about literally anything, we hopefully have an answer for you. So again, feel free to come talk to us after the session. You can also email us if you email us, it'll come to me. But there's our ortadmin@uhn.ca where we'll be able to answer some of those questions. As well, we have a booking system where you can book a one-on-one meeting with us, a 30-minute meeting when we can talk those through, depending on the nature of your concern. Happy to address it in all of these different ways. Now, so once you've been fully onboarded to UHN and you're set up and you're ready to go, I just wanted to share a few tips and tricks on mastering kind of the basics about research. So before you dive into the research, there's a few different things that are really important for you to know and kind of grasp and wrap your head around before you get started. So one thing is really important is that you're understanding the research field that you're in. So things like understanding the background information, particularly like what are the questions that led to the research that you're doing now, or what, some of the background information that established the project that you're working on now. Are there any specific findings that led to the research question that you're helping to answer? Good places to start with that are looking at review papers. These are really great papers because it helps give you broader context of the field and kind of depending on when the paper was published, but as current information as you can about that field of research or topic, I guess. As well, looking and thinking about the theory in terms of, especially if you feel like you're working in maybe some more of the wet labs or any of those kind of hands-on type of research. The theory behind the methods or the techniques or the protocols that you're doing is really important. I remember one of the kind of things that really clicked and made research really exciting for me when I started was that when I was working in a wet lab and doing a lot of like those bench side techniques and remembering and kind of connecting those back to the lectures that I were taking and learning about in undergrad. I found that really cool because you learn about it and you're sitting in the classroom and they're telling us about it. But then once I got into the lab, it's like, oh, this is actually how it works, or I'm actually doing what they're telling me. I found that really cool and kind of one of the things that got me really excited about research. So putting some effort into thinking about those theories is a cool thing when you get started. Other thing is if you're working in labs or you're involved with different hazards, just being aware of some of the safety protocols and the safety around some of those different reagents or hazards and just making sure you're following those proper safety protocols. Again, going back to like some of those basic theory and the basic knowledge, chatting with your coworkers, chatting with your supervisors, asking for some guidance on some of those foundational papers that you should look at or reviews or anything like that. They have their wealth of knowledge, so a great group of people to ask. And then once you've identified these papers, save them and read them again, especially like as you're getting more comfortable in the research that you're doing and in the topics. Going back and reading them again can really help kind of get that thought process going and kind of get you thinking even further and deeper into the work that you're doing. And I think if not the biggest thing I hope you take away from this is just ask questions. It's a time of learning. It's a time of discovery. Research is all about questions. So ask tons and tons and tons of questions. So just to build off of some of those different things that we talked about, to get those papers and get some of those resources that you need, the UHN does have a library system, and this is a great place to go for accessing all of those journal articles and papers. For those of you who still have access to your university libraries as well, that's another great place to go and check out, because perhaps some of the access might be different. You might get more from one library or the other. So maybe by checking out both of them, you have access to a ton of different papers and resources. So please utilize that as much as you can. The UHN library also hosts tons of different workshops, and they record all their workshops. So you can, learn about different research, search engines like PubMed or Medline, as well as different reference managers like EndNote and Mendeley. Those are really cool for when you're saving your papers and for citations and stuff like that. So different really cool things to go check out. For those of you involved with clinical research, there's something called the UHN Clinical Research Hub. It's a SharePoint page, which is basically just an internal UHN website. So it has a ton of different information to kind of help you do some of that clinical research. So things like provides you an overview of the study life cycle and different resources to help you at every step of that clinical research life cycle. They also have a virtual learning library. So there's topics like recruitment, if you're doing any patient recruitment, getting informed consent, research integrity, ethics, risk, a whole bunch of different things. Really, really important and critical things to do and know about when you're doing clinical research, but really any research generally, as well as relevant policies or document templates. As well, we have a department called UHN's Research Quality Integration. So this is a department involved in, as the name suggested, research integrity or quality integrity. So it's, as researchers, again, it's all of our responsibility to conduct research with integrity and to a high standard. So our QI website contains guidelines on researchers' responsibilities, again, different policies, and then how to report research misconduct, just in case if something like that were to happen. As well, they do provide consultations on research integrity concerns. So if there's ever any concern about research integrity, misconduct, if there's concerns about authorship, this is a group that you can go to ask all those questions, and then they can help guide you through some of those processes in case there's some concerns there. It's a great department to be aware of. I guess a little bit of plug to our next session on June the 12th. We will be hearing from two members, Christine and Michael, from this department, and they'll take you through all the do's and don'ts about conducting research with integrity. So it'll be a great foundational workshop for people getting started in research and things to consider. All right, so again, shifting gears a little bit now, thinking a little bit about setting goals and aligning your expectations. Another really important thing to do when get starting in research. So one of the next steps and the biggest steps in research productivity is to set clear goals and aligning expectations with your mentor. So aligning expectations between you and your supervisor or your mentor can be crucial in ensuring that you are on the same page about the expectations, which really opens the communication and improves and increases your productivity. So there are many things that you may want to discuss with your supervisor when you're getting started. So there's a lot of different things as simple as what are my hours that I'm expected to work? Where am I expected to work? Is there any opportunities if there's some days I can work at home? I'm expected to be on site every single day? Those types of things are really important to kind of figure out early on. A lot of groups and labs have meetings on a rotating schedule, one-on-one meetings, a whole different type of meeting schedule. So what meetings are you expected to attend with whom? What are the best modes of communication? Is it email? Do they have group chats, Slack, Teams? Where's the best place to get into contact should something happen? How often will you meet or have these check-ins? So again, going back to those meetings, what are the frequency of those meetings? What do I need to do to prepare for those meetings? Another important thing is in terms of research data management is where do you store your data? Is there servers? Is there hard drives? Where's the best place to keep all the data that you're generating? And then another important thing is to think about what are the expectations and the timelines for finishing different tasks. So again, working with them to understand in your learning process of what are the expectations in terms of all of these different things. Just a couple of examples here, but if there's, again, having those meetings and having those conversations will help and make things nice and clear for you as you're getting started. And then as well, the other things to continue discussing is kind of maybe at a kind of a bigger picture level and just thinking about your goals and what do you want to accomplish? What do you want to get out of your experience here over the summertime? It's a great time to learn tons and tons of things, build tons of skills and all of them, and so much more. So thinking about what do you want to accomplish? What do you want to learn? All right. So now just I wanted to share a few more resources here in terms of different training opportunities, as well as some clear career exploration opportunities. One of the big ones that I want to plug is, of course, the Office of Research Trainees, which is my department. We host a ton of different workshops, events on a whole bunch of different topics, a lot of events on research training. So over the summertime, you may see workshops coming out, which they haven't been advertised yet. Stay tuned for like things on statistics or scholarship writing. Those are some different topics like that would come out, as well as a number of different career explorations. We have a ton of career panels, workshops on specific careers to help you kind of build those skills that are important for you while you're at UHN doing your research, but also what do you want to do next? So one of our major aims. Something else that we do all the time is we try to keep a nice open communication channel. So we send a weekly email with links to both our own training opportunities, but as well as external training opportunities that we think would be relevant to the community, as well as any funding opportunities. So because you're all in this room, you received an email from me, I think, at some point, or from the ORT, which was me. So on Tuesdays, you'll receive this email, and so I do encourage you to check that out and attend both, of course, the STAR program, but then any other training that you think would be of interest to you. We also have a funding database. Again, that might be if there's any future graduate students or postdocs in this room, perhaps that might be a little bit more relevant there. But again, any upcoming opportunities to help fund your research, provide stipend support, travel costs, travel grants, we have a database for that as well. I just wanted to also note, just because I include the little asterisks here, a lot of our events are virtual and are open to both people within UHN, but also externally. So if once you're no longer part of UHN, a lot of these opportunities are still available to you. So I do encourage you to take part in them after your training at UHN is over. Another really, really, really amazing opportunity that UHN has for everybody and anybody part of Team UHN is something called Percipio. And it's a really, really cool, I know I'm hyping this up, so hopefully you think it's as cool as I do, but it's a platform that has tons of different free self-paced learning opportunities, and which provides certificates at the end of each of these different modules. So it's really great for those micro-credential learning and great lines on your CV. And because you're part of UHN, it's free. So they have courses on like communications and presentation skills, things like data visualization and management, productivity and collaboration tools, as well as leadership training, leadership essentials. So again, there's certificates that you can work towards on project management, excuse me. There's some Microsoft Office training. It's really a fantastic tool and it's free. So that's why we really like to plug this and really like kind of shout it from the rooftop because it's really a great opportunity. One thing that I particularly love and I'm in the process of working on is through Percipio, they do offer the free educational components to the project management certificate. So project management certificate is like a recognized training through the Project Management Institute. It's really expensive, but because you're part of UHN, it's discounted because you don't have to pay for those educational training because it's offered free through Percipio. So again, I encourage you to have a look through this to see if there's anything of interest to you and utilize it to your fullest ability while you're here. So you can access this by going through the My Learning, where you did a lot of your trainings kind of getting started. There's a big, huge button at the top of that page to click to navigate towards Percipio. If you can't find it, we'll link that in the email. It's also important too while you're here, just for your own professional development, career development, is just meeting people. Here is a great forum to talk to people and meet new people. Maybe, might be a little bit hard today because there's so many people in this room, but also a great opportunity to meet somebody new. We don't have any formal networking after today, but I encourage you to talk to the person next to you if you don't know them. Try to meet somebody new. Come talk to us because I recognize a few people in here from last year, but I've not met a lot of people. So again, it's great to expand your network and talk to some cool new people. Other opportunities just to keep in mind are if your departments or institutes or floors in your research building have journal clubs. If there's any seminars at your institutes, go to those. There's a lot of people that attend those. Um, all kinds of those different events, again, like we promote in our newsletter. And there's also a group called UHN STEM Pathways. So they do a lot of outreach if you're interested in, spreading the joy of science to different groups in youth. And so they're another really great group to check out. So there's a QR code to that group as well. Now, I guess we're doing pretty great on time. So before we pass it over to our grad students for our research showcase, where again, we'll be able to showcase the different research and the diversity of research happening at UHN, are there any questions about anything? We have a few minutes before I pass it over. No? Thanks for coming. If there is a chair, should anybody want one? Okay, I think what we'll do now is just pass it over now to Madalena, who will kick us off, but we'll be hearing from two graduate students and two postdocs who will tell you about the research. So it's here.
Started. Hi, everybody. Happy Friday afternoon. My name is Madalena Lugas, and I am a PhD student, but actually now a PhD candidate here at UHN. I've been at UHN for the past 5 years. I did my undergrad here at University of Toronto, where I majored in human bio and environmental bio. Then I proceeded to do my master's degree over at Toronto Rehab, which is 2 hospitals over. And then I loved it so much, I decided to do my PhD. So I am a rehabilitation health services researcher, and I study social connections and the relationship to quality of life and to understand how they impact brain health for people living with dementia. Okay, so the only thing I'm going to say, and this is kind of like my PSA to all of you, is I know most of you are probably here because you want to attend med school. Am I right? No, yeah, a lot of you probably want to become medical doctors. That was me too 5 years ago. And then I found research. I studied real people, and I kind of fell in love with it more than I ever thought I would. And here I am doing my PhD. So rather than just being the star and trying to build your CV, I really encourage you to self-reflect of why you want to go to med school. It has to be simply because I just want to make money. Really internally reflect and ask yourself why you want to help people. And I think you'll find your purpose as you navigate the STAR program and future research opportunities. It'll become a lot clearer about why you're doing the work you do. Okay, so as I said, I'm at Toronto Rehab, which is just 2 hospitals down the street. I'm a health services researcher. So what does that mean? Essentially, I look at health policies in Canada, and I understand how they do meet or do not meet the needs of people living with dementia. So specifically looking at how, for example, factors such as sex and gender, the type of dementia people are living with, where they reside in Canada, so being in a rural environment versus an urban environment, their education levels, their income, how that influences their opportunities for connections and access to healthcare services. A lot of my work has taken me internationally. So rather than just look at how Canada's health policy and national dementia strategy meets the needs of those people, I've traveled to locations such as Spain. In the summer, I'm going to the Dementia Village in the Netherlands to understand how international policies are either doing better or learning at what I can learn from those to bring those strategies to the Canadian policy context. Okay, so I mentioned the word dementia. Who here knows what dementia is? Yeah. So dementia is an umbrella term to describe a certain level of cognitive impairment. So cognitive impairment, what is that? It's when people have trouble remembering, learning new things, concentrating, or making decisions that affect their everyday life. So dementia is essentially a level of cognitive impairment where your everyday activities of living are affected. So you're having trouble bathing, dressing, eating, that sort of stuff. So cognitive impairment often occurs as people get older, but can also happen in young people. There's such thing as young onset dementia. And it's not just the same thing as getting older and forgetting certain things, it's your everyday life being impacted. And so risk factors for cognitive impairment can include family history, brain injury. So I'm sure people who play sports get concussions and that sort of stuff. That brain trauma contributes to cognitive impairment, as well as the physical activity levels and other diseases. Now, my specialty within this is social connections. So essentially what I've learned from my research is that social connection actually has a bigger effect on somebody's morbidity and mortality than things such as eating healthy and regular exercise. So having bad social connections and social connections not meeting your needs is actually more detrimental to your health than just having a healthy diet or regular exercise. So form good relationships, everybody. Okay, so this is just comparing Alzheimer's and dementia. We can go through this. And so as you can see, on the left side of the brain, you have a normal brain, which is very like spongy. But then as people experience cognitive impairment and dementia, you can see the brain shrinks and the actual shape of the brain changes. And that's what creates issues with everyday activities. So what do I do? Well, social connection is a broad term to describe how we connect, relate, and interact with one another. And in the literature, social connection is described as one big concept. Oh my gosh. One big concept. So essentially what my work does, it breaks it down. So it understands how people experience connection in terms of structure. So how much connections do we have? Function, do we feel supported and loved in our relationships? Are we receiving the support we need? And then quality. So are we satisfied in our relationships? Are we happy in them? And I look at how each of these components contributes to quality of life. So, for example, we have a lot of programs in Canada that target social connection, but a lot of these programs are really targeting just the structure. So it's just giving people the opportunity to be connected, and they're not meeting the function and quality components. So people are not feeling supported in the program, maybe because they're not forming meaningful connections or having the opportunities to do that. Okay, well, thank you. Are we doing questions, Jordan, or no?
We have time for like maybe one or two.
Does anybody have any questions about like getting involved in research? Or people research? This is people research. Yeah, go for it.
I was curious how you would translate.
The clinic, haha, yes. So most of my work does not take place in a clinical environment, rather it's more community-based. So what I do is I work with people living with dementia and their caregivers to understand like what community supports exist. So for example, I've done projects with the city of Brantford and we look at programs that exist for people with dementia. And then my job as a policy researcher would be to go in and see like, okay, who's accessing these programs? Because often the people accessing the programs are actually not the people who need them the most. So people who are, more well-educated, have sociodemographic factors such as higher income that enable them to access programs, and people who actually need the program probably the most would benefit it from the most aren't having access to that because of multiple barriers related just to their situation. Yeah. Okay, well, I'm going to pass it off to the next speaker. Thank you, everyone.
Next up, we have Anusha. I know she's, coming through the crowd. Over to you.
So I'm going to give quite a different flavor presentation. It's going to be pretty information dense. All of my work is with a rodent model for Parkinson's disease. What I want you to take away is not any of like the content, but more of an appreciation about the process and the kinds of techniques that you might be exposed to if you're choosing to do preclinical research. So I work in a stress research lab, but I have a Parkinson's background. So in my lab, we're actually studying the link between stress and Parkinson's through a structure in the brain called the subthalamic nucleus. What we know about Parkinson's disease is that it's a movement disorder that's caused by a selective degeneration of a population of neurons in the brain, and that stress can actually make this degeneration worse. So you can appreciate that in the right panel that there's actually fewer staining for neurons than in the left, and that is an animal that went through a stress protocol in addition to the Parkinson's lesioning. But we don't really understand why the two are linked or the mechanism in the brain that represents the intersection. But what we think is that it might be happening through a brain area called the subthalamic nucleus. This is a region of the brain that is affected in Parkinson's disease, leading to movement related symptoms. And it's also a target for treatment using deep brain stimulation. It houses a population of cells that express a stress responsive neuropeptide called corticotropin releasing hormone, or CRH. But beyond that, we don't really understand why this potentially stress responsive group of neurons is located in a disease relevant structure in the brain. So that's really where my research has been focused on, is characterizing this population of cells. I use a technique called patch clamp electrophysiology, which is a really cool way to measure neuronal activity because all of these cells are electrically active. So we can actually tap into the circuit and listen in to what those neurons are doing on an electrical level. So I dissect out brains from mice and keep that tissue alive. I prepare sections of my brain slices and incubate them in artificial cerebrospinal fluid. And with this method, you can keep those neurons alive for about 8 hours outside of the body. So those slices look a little something like this in a cartoon form, and I'm able to directly access single neurons using a glass pipette that's connected to an electrical circuit. And I can record activity that looks a little something like this. In practice, this is what I'm looking at down my microscope. That oval-shaped large structure is the subthalamic nucleus of a mouse. And you can also see that there's a glass pipette poking into the top half of that structure. So that's me patching onto a single cell. And I obtain real recordings looking like this. So the point of these experiments is to establish baseline activity, which I can then compare to a stress state. So do these measures again in a mouse that went through a stress protocol, and then compare them to mice that have been induced to mimic Parkinson's disease. So this comparison has led me to analyze these recordings. So I just want to give you some appreciation of how that looks. So each individual vertical spike that you see there is an action potential. It's kind of a single new unit of neuronal communication. And we can induce this by injecting different levels of current. So all of these square pulses are overlaid on top of each other, but in reality, I'm going through each of these individual steps one at a time. So what I'm doing here is just counting the number of action potentials in response to the current injection. And what I have observed is that in wild-type mice, which is a term for control animals, I see that the number of action potentials generated is actually increased when these animals are exposed to stress. So stress seems to make this population of neurons hyperactive. And that pattern seems to be opposing in a Parkinson's model, at least in my preliminary evidence. So I don't want to get too fixated on this particular data point, but I wanted to show you examples. The impact of this kind of work is that we're establishing mechanism in a much more accessible model in a mouse, but at the end of the day, we do want to relate this back to human brain activity. And by doing so, we can drive the understanding and development of new drug targets, potentially through the CRH-dependent pathway. We can better understand existing treatments for what they're actually doing in the brain, because right now it's kind of a shot in the dark. We know that it works, but we don't know why. And finally, because my research implicates stress, it also emphasizes the importance of stress reduction, not only in a disease state, but for general improvement of quality of life. So I'll end my presentation there and maybe open up for questions.
We've got lots of time, so if you have any questions, please feel free.
Over this question, but how did you get started working with mouse models as opposed to
Yeah, I went through the traditional research pipeline. I did an honors project in my 4th year where I started working on this electrophysiology technique, but I learned that in rats. And then I switched to a lab that works with Parkinson's, and so I was doing more mouse work there. And so it's been a continuation of that line of work. But this is the first time I'm actually working at an institute where I'm so close to similar experiments in people at the same time. So hopefully I'll have a chance to intermingle more with the clinicians and fellows that are doing similar kind of research in people.
People about inducing the Parkinson.
Yeah, so they can be induced in a number of ways. Our particular model is called the 6-OHDA model. So 6-OHDA is the name of a neurotoxin that gets injected into the brain. So it causes a very similar pattern of degeneration that you would expect to find in postmortem tissue in people. But it's not a perfect model, but it is a good way to, like, quick and dirty, get it done, so that we can then go into the subtle differences later.
What are your thoughts on what's the human brain tissue?
Yeah.
Like, could that translate better with like human bodies?
That would certainly offer like an intermediary step. There are some technical limitations to how these organoids are created and how they mimic a human brain, because it's something that's sort of derived from adult tissue, obviously. Sorry, not obviously, but usually. So what happens is that it doesn't fully represent a human brain. But this particular technique has been used. You just get a lot of variability with organoids. So the mouse model and like total brain activity in humans tend to be less variable than the brain organoids in the middle. Yeah. I looked initially in my cohort and I didn't see any sex differences in the wild type animals, but I don't have enough ends or sample number to be able to distinguish, but the end goal is to have enough data to actually talk about that too. It is something important to the disease state in people, is that you get different proportions of presentation and age of onset. So it's really important to know those things. Yeah, thank you.